You already heard, and you all know that pancreatic adenocarcinoma, PDAC, is one of the most aggressive cancers with a 5-year survival of only 6%. And in the last 20 or 30 years, I think it remained one of the most lethal cancers, and there's been very little advance in the treatment of pancreatic cancer. And although I'm a firm believer in the power of immunotherapy, and I think that's probably going to be the only way to treat cancer in the future, I also believe that we need to think a lot more about prevention. And prevention research actually has not been adequately funded in the US, and I think quite a few cancers can actually be prevented. And both pancreatic and liver cancers are among some of the more preventable cancers if we understand how all the risk factors interplay to increase the development of these cancers. And it seems really that it's really a lifestyle that is resulting in this increase incidence of pancreatic cancer that we have seen in the last 20 years or so. And I think, first of all, we are living longer. And second, we have some tendencies to abuse things such as alcohol and tobacco, which can increase the risk of pancreatic cancer. So one thing that we need to understand about pancreatic cancer, which is rather unique, is despite being a major killer, it's still a rather rare cancer. And the overall lifetime incidence of PDAC is only about 1% compared to 10% for colorectal cancer. So if we are thinking about early diagnosis, early screening, it's going to be quite expensive, and because we're trying to really identify a very rare cancer compared to a much more common cancer such as colorectal cancer, where we do have effective screening. The other peculiar thing about pancreatic cancer is that Paning 1 lesions, which are the precursor lesions that lead to the development of PDAC, are actually rather common and are detected in about 70% of individuals above 70 years of age. And yet, most of these individuals, not only because of their advanced age, but because of other factors, will never develop pancreatic cancer. So we think that the key to understanding how pancreatic cancer evolves is to really understand how these quiescent Paning 1 lesions, which will never really cause any trouble in most people, decide one day to progress and develop into full mature and full-blown aggressive cancer. So if you think about how cancer evolves, it's very clear now from cancer genome sequencing that you need many different mutations to generate cancer. And a single mutation, especially a KRAS mutation, is insufficient. So let's think about a cell in the pancreas, an acinar cell that is acquiring a KRAS mutation. That cell will never progress to cancer unless it will acquire an additional mutation, for instance, inactivation of P53. But even that is not sufficient based on human cancer sequencing. And then you need another mutation, for instance, loss of P16. And only when you accumulate these 3 right mutations in a single cell type, this cell type will win the cancer jackpot and will be able to progress to cancer. However, it's really not that easy, because if we think what is the main driver of oncogenic mutations, and one likely possibility are reactive oxygen species, and once a cancer cell, a precursor cell, acquires a KRAS mutation, it has many different choices, and it can undergo senescence, and that's usually what happens with Panin 1 lesion, and that's why they're so abundant. The cells can either die in response to bombardment with reactive oxygen species, which are very toxic, and only on rare occasions, the cell will acquire the capability to divide and accumulate additional mutations. So we really need to understand what allows the cells that are continuously exposed to oxidative stress to be capable of surviving in such a harsh condition, then dividing, acquiring additional mutations, and progressing to cancer. And we speculate from various studies that there is one vicious transcription factor that was thought to be protective from cancer, but we think it's actually the key to cancer progression, and that's a transcription factor called NRF2, which is an activator of the antioxidant response. So we have postulated that PDAC risk factors, such as obesity, alcohol abuse, smoking, chronic inflammation in the form of pancreatitis, as well as old age, are responsible for the progression of the quiescent Panin 1 lesions to higher grade Panins, which are proliferative lesions, and then they can make it all the way to full-blown pancreatic ductal adenocarcinoma. And we think that all of these risk factors may operate through a common mechanism, and that's interfering with autophagic protein degradation. And even though interference with autophagy is known to kill tumor cells, cancer cells, and can also induce ROS accumulation in normal cells, actually in the cancer-initiating cells, it's something that increases their ability to survive and proliferate through the activation of NRF2. So I don't think I need to spend much time describing what autophagic protein degradation is. It's a very important safety mechanism that gets rid of our cells of misfolded proteins and damaged organelles such as mitochondria that produce reactive oxygen species. And it's also very important for cells to age properly and maintain their function throughout life. But the problem with all of the risk factors involved in both the development of pancreatitis and the progression to pancreatic cancer is that many of them can interfere with proper autophagic protein degradation at different steps, both at the initiation of autophagy as well as the termination of autophagy. Alcohol will do this, nicotine and other components in cigarette smoke will do this, old age will do this, and obesity will do the same. And defective autophagy will result in cell damage. Acinar cells that are damaged will give rise to pancreatitis, and then through the inflammation in the pancreas, you further increase the risk of progression to pancreatic ductal adenocarcinoma. So we wanted to see actually how disturbance in autophagic protein degradation would lead to development of pancreatitis first, and then understand the role of pancreatitis in the development of PDAC. And a few years ago, we made the unexpected observation that if we knocked out IKKa alpha in pancreatic epithelial cells, the mice that lacked IKKa alpha in the entire pancreas developed spontaneous pancreatitis. There was increased acinar cell death, increased immune cell infiltration. There was compensatory proliferation of the surviving cells, trying to compensate for the loss of acinar cells. And then you see quite clear onset of fibrosis, and eventually the mice develop a highly fibrotic tissue that looks very much like human chronic pancreatitis. And what we found was that even though IKK alpha is an important regulator of NF-kappa B activation and inflammatory responses, that its role in pancreatitis was actually mediated through its involvement in the completion of autophagy, and had nothing to do with its protein kinase activity, activation of NF-kappa B, and the entire response is NF-kappa B independent. And since we suspected that IKK alpha is doing this by interfering with autophagic protein degradation, we decided to really knock out autophagy at the very beginning and delete a protein called ATG7, which is very important for the initiation of autophagy, and see whether our hypothesis that disturbance to autophagy can lead to pancreatitis through this rather simple experiment. So we knocked out ATG7 in pancreatic epithelial cells, and we got a similar but actually much more severe phenotype to the knockout of IKK alpha. Again, there's a rather extensive acinar cell death in the ATG7 knockout mice. You see quite strong compensatory proliferation. There are inflammatory infiltrates, which are not shown here, and you get very, very extensive fibrosis of the pancreas. So again, either interfering with the completion of autophagy or interfering with the initiation of autophagy, you end up with pancreatitis. And when we looked at closer detail using electron microscopy, exactly what happens in the acinar cells that either lack IKK alpha or ATG7, we found that the cells undergo ER stress, and in addition to ER stress, they accumulate quite a lot of abnormally looking mitochondria. And trying to understand how defects in autophagic protein degradation can trigger ER stress, we decided to focus on a protein called P62. P62 is a very important chaperone for damaged proteins. It takes them into newly formed autophagosomes for clearance. Itself, it is degraded through autophagy, and anytime you interfere with proper autophagy, P62 will accumulate, and instead of causing the degradation of ubiquitinated proteins, it actually binds to them and lead to formation of protein aggregates that now actually cause ER stress instead of letting the cells get rid of self of all this damaged protein. P62 is also a signaling protein. It can lead to the activation of NRF2, which I mentioned earlier on. It can also lead to the activation of NF-kappa-B. And as far as autophagic degradation is concerned, it's only 5 of different autophagy receptors. So if you delete P62 rather than accumulate it, there's actually no defect in autophagic protein degradation whatsoever, because there are actually quite a few other autophagy receptors that can compensate for the loss of P62. So we wanted to see if P62, which accumulates in both IKK alpha knockouts and ATG7 knockouts, would actually prevent the development of ER stress and will prevent all the pathological consequences of that, and will prevent the development of pancreatitis. And indeed, if you generate an additional knockout of P62 in the IKK alpha pancreatic knockout mice, you completely reverse the phenotype, and the mice have a perfectly normal-looking pancreas. They don't develop any pancreatitis, no fibrosis whatsoever, no inflammation, and everything is fine. However, if you knock out P62 in the ATG7 knockout mice, you only compensate a little bit for the loss of acinar cells. There's a small increase in amylase-producing cells, but there's no reduction in ER stress markers. The tissue is still ER stressed. You get some reduction in NRF2 activation, but you really don't block the development of pancreatitis in these mice because of the very strong ER stress. And then we looked at P62 in other tissues, and a tissue that is actually quite related to the pancreas, both in developmental origin and also in many of its function, is the liver. And it turned out that if you knock out P62 in the liver and you conduct quite rather extensive transcriptomic analysis, we found there's only one pathway that is totally dependent on the expression of P62. And again, this is the NRF2 antioxidant response. And when we looked at human hepatocellular carcinoma, where NRF2 is activated, again, we found very strong correlation between the accumulation of P62 and the activation of the NRF2 response. And from our studies on hepatocellular carcinoma, we have postulated that P62, which accumulates in all chronic human liver diseases, and I'll show you it also accumulates in human pancreatitis, as well as PDAC, is leading to liver degeneration and, more importantly, to the development of hepatocellular carcinoma through its ability to titrate a major inhibitor of NRF2 activation. It's a ubiquitin ligase called KEAP1, and when KEAP1 is titrated, NRF2 accumulates, and by activating the antioxidant response, it actually does not protect cells from oxidative stress. It actually allows cells with oxidative stress to survive, and once they survive, they accumulate additional mutations and eventually give rise to cancer. So we wanted to see what else is happening upon the ATG7 ablation in the pancreas. And basically, we found that the loss of ATG7, the loss of autophagy, results in activation of three protective responses or compensatory responses. First of all, you see accumulation of reactive oxygen species as a result of the ER stress, but this leads to activation of NRF2. NRF2 protects cells from ROS-induced death, but it doesn't protect them from ROS accumulation. You see activation of AMPK because the damaged mitochondria cannot produce enough ATP, and activation of AMPK results in inhibition of protein synthesis, which is one mechanism to reduce ER stress, but it's not very successful. And then you get compensatory proliferation of the residual acinar tissue in the pancreas, both through direct proliferation and through also acinar ductal metaplasia, which is a normal process used by the pancreas to repair damage. But as I'll show you, acinar ductal metaplasia is also a very strong tumor-promoting response and is involved in the progression from pancreatitis to PDAC. So equipped with all this knowledge gained from mice, we decided to look at what happens in human pancreatitis. And we and other groups found that in human pancreatitis, there's very strong accumulation of P62. I'm not going to show you this here. And then we decided to look at pancreatic cancer and what actually happens. So actually, the data from the chronic pancreatitis is shown here. You start seeing accumulation of P62 in chronic pancreatitis. These are just representative samples. We pretty much see it in every single sample. You see activation of NRF2, both increased expression of NRF2 itself, and it's a lot easier to note that the expression of NQO1, which is a classical NRF2 target genes. If you look at Panin lesions, these are actually advanced Panin lesions isolated from tumor-bearing pancreatic tissue, you can see that in the panins, there's also strong accumulation of P62, even stronger activation of NRF2, and you see further progression in PDAC, where P62 is expressed in all the tumor tissue, not in the tumor microenvironment so much. And you can again see that NRF2 is activated in the epithelial cells of the tumor, in the carcinoma cells, but not really in the stromal fibroblasts that are so rich in these tumors. So we wanted to see what P62 actually does in human pancreatic cancer. We knocked it down in MIA Paca-2 cells, and using shRNA, we implanted the cells into immunocompromised mice, and you can see quite clearly that when you get rid of P62 or reduce its expression, you reduce tumorigenic growth in this very simple model of xenograft formation. If when we knocked out IKKa alpha, we increased the expression of P62 in the MIA Paca cells, we made them more tumorigenic. But if we knocked out again P62 on top of the IKKa alpha, we brought the cells back down. So clearly, this axis between autophagy, P62, and so on controls tumorigenicity in human pancreatic cancer, not only the progression of the disease. And we decided to see whether P62 is indeed operating through NRF2, as we believe it does in the liver. And knocking down NRF2 with shRNA generated a very similar effect to knocking down P62 itself. Again, reduced tumor growth in vivo, and reduced sphere formation ability in vitro. And with the reduction in the sphere forming ability of both the P62 and the NRF2 depleted cells is related to reduced expression of a number of progenitor and stem cell markers, including ductal markers such as CK19, progenitor markers such as SOX9, nestin, PDX1, and so on, as well as general stem cell markers, cancer stem cell markers such as EpCAM, CD24. We see reduced activation of the Notch pathway and increased expression of senescence markers such as P27. And you also see effect on expression of various inflammatory mediators, which are probably all related to the effect of P62 on NF-κB activation. Now, when we switch to mice, we want to see what would be the effect of inhibition of autophagy, something that would happen upon extensive alcohol consumption, cigarette smoking, obesity, and so on. But the problem is that you cannot completely block autophagy by depletion of ATG7 as we did to generate pancreatitis, because as was shown by Kevin Ryan and his co-workers, the depletion of ATG7 results in enhanced formation of Panin-1 lesions, but these lesions will never progress because there's massive accumulation of P53, a very strong tumor suppressor, and there's no further progression from the Panin-1 stage in these animals, which only have the KRAS mutation. So you cannot use ATG7 ablation to study the role of autophagy or autophagy defects in tumor progression. So we decided instead to only attenuate autophagic degradation partially through the ablation of IKKa alpha and see whether that will enhance the development of disease in mice that only express KRAS, active oncogenic KRAS in the pancreas. So we are not deleting P53, we are not inducing deliberate second mutations, We only want to understand how you progress from the initial Panin 1 lesion to full-blown PDAC. So knocking out IKKa alpha on top of activating RAS in the pancreas results, again, in accumulation of P62, defective autophagy, activation of NRF2, and if you look at the gross appearance of the pancreas, you can see that it looks pretty messy, and you get very aggressive tumors already at 5 weeks of age in these mice. And if you look at tumor latency, there's about 20 to 30-fold decrease in tumor latency when you knock out IKKa alpha, which is not a real tumor suppressor. The mice will progress to fully aggressive PDAC by 3 months of age, and pretty much the entire mouse cohort is dead by 6 months. And before that, not only they display invasive, fully invasive PDAC, you also find metastases to different organs, but because the mice die so quickly, there's not a whole lot of metastases. So we ask what are the mechanisms that are involved, and we found that as expected and as seen in pancreatitis alone, ablation of IKKa alpha enhances acinar to ductal metaplasia and increases the formation of Panin 1 lesions. But in this case, we see the entire progression from Panin 1 through Panin 2 to Panin 3 and eventually to PDAC within a rather short time. And those of you who work with mice with only KRAS mutations know that only 10% of these mice would actually form aggressive tumors at 1 year of age. So really, these mice don't show any appreciable progression within a short time. But in the absence of IKKa alpha, you get as rapid progression as knocking out a bona fide tumor suppressor such as P53. So when you look at in further detail what actually happens in these Panin lesions as well as in the ADMs, you find that the ablation of IKKa alpha renders all these quiescent lesions in the KRAS mice proliferative. There's a huge increase in cell proliferation in the lesions, explaining how they progress. There's also decrease in expression of senescent markers, and we ask whether this effect, as seen in the pancreatitis model, also depends on P62. So we generate a double knockout mice. P62 is knocked out, IKKa alpha is knocked out, KRAS is expressed, and when you get rid of P62, you pretty much lose most of this enhancing effect of the loss of IKKa alpha. Pancreatitis is reduced, and acinar to ductal metaplasia is reduced. You mostly get Panin 1 lesions that don't progress to higher grade Panins, and tumor development is slowed down again. Reduced cell proliferation in the absence of P62, and increased expression of senescence markers such as senescence beta-galactosidase. And again, we also see increased expression of P27. The effect of P62 is dependent on NRF2 activation. You see activation of NRF2 in mouse pancreatic cancer, just like you see in human pancreatic cancer. And if you look what P62 is doing, we find that it controls exactly the same progenitor markers and the stem cell markers as well as ductal markers in the mouse pancreas that it does in MIA Paca cells grown in tissue culture. So you get rid of P62, reduce expression of stemness, reduce cell proliferation, reduce expression of proliferative genes, very similar to what we have seen before. And when we looked at stem cell formation in vitro using sphere assays and flow cytometry, again, you knock out P62 and you reduce the formation of cancer stem cells in these animal models. And whether the effect of P62 is NRF2 dependent, the answer is yes. You knock out NRF2, you get very similar effect to getting rid of P62. And when you knock out NRF2 in the acinar cell cultures from these mice, again, you see loss of stem cell markers, loss of proliferative markers, loss of progenitor markers, very similar to what we have seen when P62 is knocked out, and there's increased expression of MDM2, which is an inhibitor of P53 activity. So in addition to decreasing the expression of stemness markers, you get increased activity of P53 due to the loss of MDM2. So to summarize, what we find is that reactive oxygen species can probably cause the progression of Panin-1 lesions because they don't induce senescence and they don't induce cell death, because you get activation of this NRF2 response, which in addition to protecting cells from oxidative stress, also induces the expression of stem cell genes, probably through effects on Notch signaling, and also leads to some inhibition of the P53 response that trying to block the induction of stemness. And altogether, now these Panin-1 lesions, which are quiescent, will start progressing, accumulating more mutations, giving rise to Panin-2, Panin-3, and eventually PDAC. And hopefully, we can now start to look for chemicals that, instead of activating NRF2, would actually block NRF2 activity and may slow down the progression of pancreatic cancer. And with this, I'd like to close and acknowledge the outstanding work done by Yelena Todorich, a postdoctoral fellow in the lab, who did most of the cancer work, and Laura Antonucci, who actually looked at the role of autophagy in controlling the control of pancreas health, and the role of autophagy disturbances in the development of pancreatitis. We had very important collaborators at the Burnham Institute. Jorge Moscat and Maria Diaz-Meco provided us with the P62 flox mice. Collaborators at UCLA, Anna and Ilya Gukovsky, have helped us with the analysis of pancreatic enzyme expression. And Ralph Ruband and Michael Goggins at Hopkins have provided us with all the TMAs used for the analysis of P62 and NRF2 activation in the human tissue. And thank you for your attention.